This video serves as the pre-lab and the post-lab video for the lab called beanbag isotopes. At the beginning of the 19th century, John Dalton proposed a new atomic theory. All atoms of the same element are identical to one another and equal in mass. It was a simple yet revolutionary theory. It was also not quite right. The discovery of radioactivity at the beginning of the 20th century made it possible to study the actual structure and mass of atoms. Gradually, evidence began to build that atoms of the same element could have different masses. These atoms were called isotopes. How are isotopes distinguished from one another? What is the relationship between the atomic mass of an element and the mass of each isotope? The purpose of this experiment is to investigate the mass properties and relative abundance of isotopes for the beanbag element, which has the symbol Bg, not a real element, and to calculate the atomic mass of this element. You should construct the data table and the results table as shown in your lab manual, but we're going to make a couple of minor adjustments to the data table and to the results table. The data table has room for beanbag isotopes numbers 1, 2, and 3. It has a column for the number of atoms and a column for the total mass of the atoms. We're going to add one more cell down below the number of atoms, and in this space, this is going to be the total number of atoms for all three isotopes. Your results table has a column labeled beanbag isotope and has 1, 2, and 3 in it. It has a column for average mass, and it has a column for percent abundance. I would like to have you add a third column, but it's going to go in the middle, so you'll have a total of 4 total columns. And in this column in the middle, this is going to have the heading relative abundance. The procedure is fairly simple and straightforward. There are 3 procedural steps. The first is that you are to sort your beans into 3 different piles according to similarity, as you see in this picture here. You'll assume that each type of bean represents a different isotope and that each bean represents a separate atom. You're going to place each isotope group, each bean type, in a separate weighing dish. You're going to count each of the 3 batches of beans, and you're going to record that number of beans, which we are going to represent as individual atoms. So each one of these beans represents a different atom of that particular isotope. So you might want to say that the red kidney beans are isotope number 1, that I think these are pinto beans, we'll say are number 2, and the navy beans are isotope 3. So you'll want to count how many atoms are in group 1, how many isotopes or how many beans are in group 2, and how many beans or how many isotopes are in group 3, and record it in your data table. The next thing you'll do is measure the total mass of each of the beans, of each of the type of beans, or the Bg atoms belonging to each isotope group. You'll record that mass to the nearest 0.01 grams in your data table, and here's the note to tare the balance with an empty weighing dish and then add all those beans of that one type. You've done this before, but this is essentially what it should look like. So make sure that you put the empty weighing dish on the scale. Then you'll hit the re-zero button, which is the on button. You'll put the beans into that weighing dish, and then whatever number pops out onto the display, that's the mass of that particular isotope. You'll record that mass in the data table where it says total mass of the atoms. All right, so at this point you should have the procedural steps done. That's essentially it for the lab itself. This part of the video will talk about how to treat your data. So this is the post-lab portion of this video. The first post-lab question says we're going to determine the average mass of each Bg isotope to 3 significant figures. and we will enter the results in the data table. So when you go to do the postlab questions, we're going to start by writing the postlab questions as our heading, and we're going to be starting with question number 1. And when in chemistry this year, when a postlab question is a calculation, you need to write a heading that describes what the calculation is trying to accomplish. For this question, what we are trying to find is the average mass of each isotope. To do that, for bean type 1, you will take the total mass for that particular bean type, get that from your data table, and you will divide it by the number of atoms of that particular bean type. That will give you some number that represents the average mass of that isotope, so that should be in grams. You'll do this again for bean type 2 and bean type 3, and you will get 3 masses. You will then take those masses and you will record those answers. So this answer that you get here belongs here. The answer for number 2, isotope 2, goes there, and isotope 3 will go in that spot right there. Okay? So that's postlab question number 1. Postlab question number 2 says, what is the total number of beanbag atoms in the original sample? Calculate the percent abundance of each isotope. We're going to divide the number of atoms of each isotope by the total number of atoms and multiply the result by 100. Enter the results to 1 decimal place in the results table. So what we're going to do is we are going to divide this problem into 3 different parts. So this is question number 2. And in question number 2, part A, we're going to find the total number of atoms in each baggie. And this is where you'll show the work that you have recorded in your data table. It should be the number of beans in number 1 plus the atoms in number of beans in sample number 2 plus the number of beans in sample number 3, and you'll have some total number of total atoms. For part B, we are going to find the relative abundance of each isotope. To do this, you will take the number of beans that you have, the number of atoms from sample number 1. Right? That number would come from your data table, and you will divide that by the total number of atoms that you just calculated in part B. Okay, you're going to get some decimal number. Your answer here should be some decimal number. That is your relative abundance. Okay, there is no unit on this number. It's just a relative abundance. You'll do this for number 2 and for number 3. So except here it'll be the number of atoms from bean type number 2, divide that by the total number of atoms. You'll get some relative abundance, and you'll repeat that for bean number 3. Okay, the answers you get there, your answer to that first problem would belong in that column that you had labeled as relative abundance. That'll be A. Your answer here goes here, And the third one that I didn't show you as an example will go here. To continue answering question number 2, this is now part C, where we are calculating the percent abundance of each isotope. What you will do here is you will take the relative abundance for bean type number 1 and you multiply it by 100. You'll get some number that is a percentage. You'll continue that for beans type number 2 and 3, and you will have 3% abundances. And these will go here. So your answer for isotope number 1 will go where I have the E's labeled, the F, and the G. That's where those numbers will go. Postlab question number 3. The atomic mass of the beanbag element represents a weighted average of the mass of each isotope and its relative abundance. We're going to use this equation that follows to calculate the atomic mass of Bg. To do that, we will divide the percent abundance of each isotope by 100 to obtain its relative abundance. We do not have to do that because we have already done that, so that part may be skipped. So what we're going to do is we're going to take to find the atomic mass, we'll take the relative abundance from isotope 1, and we will multiply it by the mass of isotope 1. And we'll add that to the relative abundance of isotope 2 times the mass of isotope 2, and we'll add that to the relative abundance of isotope 3 times the mass of isotope number 3. So let's take a look and see how you would do that particular problem. So this is postlab question number 3. We're going to label that as the atomic mass of the Bg beanbag element. And we're showing how we're doing our calculation, which is atomic mass is equal to the relative abundance of isotope number 1 times the average mass of number 1. Both of these numbers can be found on your results table. When you multiply those together, you will get some number that you'll record there. You'll repeat this process for isotopes number 2 and 3. And you can just substitute in the numbers. You don't necessarily have to write in all of these things in parentheses. I'm just giving you reference points of where you should be going to get these numbers. So relative abundance of number 3 times the average mass of number 3, and you'll get some number. You will then add those 3 numbers together, and that will be the average mass, the average atomic mass. Now this mass unit is in grams, so please make sure that you record that unit. To follow this lab, I'd like to have you write a summary. Notice summary is spelled with an A, not an E. And your summary of procedures should explain to me as a story, as a sentence, 2 sentences, 3 sentences, what did you do with the beans? Tell me what you did with the beans. You sorted them, you counted them, you weighed them. And from that information, you then were able to calculate atomic mass. Right? That's essentially the heart of what the procedure was about. We're going to follow that up with a conclusion statement. And in this conclusion, I would ask that you could write these words, and you can just fill in the numbers as you found them. So you can say, in this lab, we found the percent abundance. Please spell the word percent, don't give me the symbol. In this lab, we found the percent abundance of brown beans as some percent. Whoop. For speckled beans as whatever percent abundance you found there. And white beans, I suppose white doesn't have to be in parentheses. I suppose none of those have to be in parentheses, but and white beans as whatever percent you found that to be. From this information, the weighted average was calculated as some number of grams that you just calculated as your postlab question number 3, which represents the atomic mass of the element of the Bg element. All right, that concludes the pre-lab and post-lab video for the lab called Beanbag Isotopes.